Hi everybody, this is the last mini lecture of chapter 15. We're going to talk about drug resistance and then proper antimicrobial drug stewardship, so the use of antimicrobial drugs properly. By the end of this, you should know what antimicrobial resistance is and be able to compare both intrinsic and acquired resistance. You should also be able to name and describe the three categories of acquired antimicrobial resistance. And then I'd like you to be able to discuss the use of antimicrobials selected for resistant microbes that we use for resistant microbes, and then summarize how the resistance may spread by agricultural and clinical misuse. The last thing that I want you to look at is what can be done to reduce the emergence of antimicrobial resistance, and also talk about why it's so difficult to develop new antimicrobials, especially antibacterial or antibiotics. So pathogens, and in this case we're really talking about bacteria, can have intrinsic and or acquired antimicrobial resistance. And antimicrobial resistance just means that a microbe is not affected by a drug therapy that is intended to inhibit or eliminate the pathogen. And resistant microbes are what we call superbugs. So when a pathogen has intrinsic and or acquired antimicrobial resistance to a particular drug, they can cause super infections. And so the way that this can develop is that in any microbial population, so in this case, think about maybe all the microbes that live in and on you. If we give a drug, keep in mind that in this huge population, remember there's going to be billions upon billions of cells of bacteria in and on us. Invariably, there will be a few that have resistance to a given drug. When we then give that drug, what will happen is that the ones that are susceptible will die off, and the ones that are not susceptible will survive. And what happens when these bugs can survive? They can reproduce. And so those resistant microbes then can thrive because there's no competition. All the susceptible ones have died off. And these resistant microbes can also share those resistant tools through horizontal gene transfer. We're going to do a quick refresher on horizontal gene transfer. Hopefully you remember that from our genetics chapter. So, when we think about this, the problem of these resistant pathogens started right after antibiotics became widely used. Even with penicillin, within a few years of it, we saw resistant bacterial strains that were discovered. And there is an epidemic increase in these drug resistance, and that's because we have largely overused antibiotics and misused it. So as I said, there's two different types of antimicrobial resistance. There's what's called intrinsic resistance, meaning that an organism was basically, for lack of a better term, born with a resistance. And then there's acquired, meaning that in its lifetime it picks up resistance. And so looking at this, the intrinsic would be things that are something that maybe prevents it from being susceptible to some antibiotics that it naturally comes with. So perhaps there's a cell wall composition that makes it difficult for an antibiotic to get in. Or in the case of ones that can form endospores, this would be something that we think of as a natural or intrinsic resistance. We can also have those acquired, and acquired means that perhaps it has a mutation that gives it resistance, or through horizontal gene transfer. Remember, we have conjugation, which is the sharing of genetic material from one living microorganism to another. We can have transformation, where a microbe can pick up new genetic information from the environment. And we can have transduction, where when it's infected by a bacteriophage, it can pick up new genomic information. So, intrinsic resistance, that natural resistance to antimicrobial drugs, this is what makes those certain pathogens hard to eliminate. So a couple of examples of that: Mycoplasma pneumoniae, it actually doesn't have a cell wall. It's one of the exceptions to bacteria. And so it's resistant to any drugs that would target cell wall synthesis because there's no target for them. Clostridium difficile is one that can form endospores because it's resistant to most antibiotics because it can hang out in its dormant stage until the antibiotics are gone. So, some other intrinsic ones are Mycobacterium tuberculosis. This one, actually, the cell wall has a lot of wax in it, so it has this mycolic acid that can actually present diffusion of drugs through the cell wall, so any that would target the inner workings of the cell would be prevented from getting in. Keep in mind gram-negative bacteria. have that outer lipid membrane, and many drugs can't get past that outer membrane. And then probably the most common or the most difficult one to get rid of is microbes that form biofilms. Remember, biofilms are those communities of different microbes that form many layered community, and often the drugs can't get past that top layer, and so those microbes that are deep in the biofilm would survive. With acquired resistance, this is where we see resistance comes from a genetic mutation or picking up resistant genes. So the three main ways that microbes can evade an antimicrobial drug is they can either alter the drug's target. So if the drug has a particular target, maybe there's a mutation that makes that target look slightly different. Inactivate the drug. This is often what happens with penicillin. Ones that are resistant to it can actually break up the drug before it can cause harm to the cell. And then the last way that bacteria can have resistance is through reducing the drug concentration within the cell. And this can either be through blocking the drug from coming into the cell, or perhaps they have a pump that can actually pump the drug out of the cell as it enters. And I have a figure showing this. So one of the things we think about is perhaps there's a protein or something or some sort of target within the cell. If this antimicrobial drug is coming to target it, if we change the target, this drug will no longer work. Reducing the amount of the drug within the cell, we can either pump it back out as it comes in, or we can make it so that it can't find a way in. And then the last way is even if the drug gets in, we actually break it down so it's no longer harmful as a bacterial cell. So why do we see such a high amount of resistance right now? And probably it's something, if you work in healthcare, you've heard about. Antibiotic resistance is not caused by the use of antibiotics, but it is, we do select for then those ones that are resistant. And so when we have antibiotics used, there's selective pressure that selects for those bacteria that can survive antibiotics. One of the ways that we see this is non-compliance with prescribed drug or dosing parameters. So when we tell somebody to take a antibiotic for 10 days and they stop early, often these ones that have slight resistance have a great chance of surviving. We can also see that many patients miss doses or they self-medicate with leftover prescriptions. And this is basically just selecting for those microbes that can survive a little bit of the drug, but not a full dose. And so this low dose exposure actually serves as selective pressure. So this is showing all the different human behaviors that can accelerate drug resistance. On the left-hand side is the ones that we think of as agricultural practices that promote resistance, and on the right-hand side are those clinical practices. So let's start with the clinical, because for most people, that's where we understand it the best. So throughout the world, there's many countries that actually don't regulate antibiotic use. You don't need a prescription, so we get a lot of overuse and abuse. We get non-compliance with the dosing of some antibiotics. And then lastly, we get healthcare settings where there are lots of pathogens, a lot of transmission opportunities, and lots of antibiotics being used. So we would see this in long-term healthcare facilities and hospitals. On the other side, which maybe people don't think of so much, we have agricultural practices that have promoted this. And that's because antimicrobials are put in animal feed. And what happens is we get resistant bacteria in the gut of these animals. Then these resistant microbes can be spread through the waste of the animals. In addition, these animals that are fed antimicrobials, when they're slaughtered, the meat can be contaminated with those antibiotic-resistant microbes, which means if you get an infection from that, it will be hard to treat. And then lastly, when we think about the plants, so think about all of those romaine lettuce recalls and different produce. When we put the manure that's contaminated on crops, what we can see is that we introduce the resistant bacteria into the food chain. It can be drawn up by these plants that can also cause a problem. So when we think about that, this is just going over that. In the agricultural setting, those antibiotics are commonly used. And in the U.S. alone, each year, there's around 26 million pounds of antibiotics that are used in animal feed. The FDA has banned adding antimicrobials in human medicine to animal feed, but there's still potential for cross-resistance. So basically, we're trying not to use the same medicine in agriculture that we're using in humans. In the clinical practice, there's a bunch of different ways that drugs are misused. Often they're used for self-limiting infections that would go away on their own. We treat them, so sometimes people come in with something that they don't need the antibiotic for. Also, sometimes they're given for non-infectious conditions. And lastly, and probably the most common, is that they're given for viral infections. And this can be a number of different reasons that this can happen. The doctor doesn't know for sure that it's viral and thinks it could be bacterial. A patient demands that they get medication for being ill, even though it's a viral infection, and there's pressure on the doctor to do that. And right now we would guess that around 30% of all oral antibacterial prescriptions are actually unwarranted. They should not be given out. And so that might be misdiagnosis, patients again pressuring for that prescription, or the unregulated use of it in developing countries. So when we think about the healthcare setting, this is the perfect place for antimicrobial resistance to grow. They're commonly found in healthcare settings, and it has a combination of that there's lots of pathogens, right? You're in a hospital, there's lots of sick people, so we have lots of pathogens present. In addition, we have tons of antibiotics that are being used, which are then in the environment, which can select for that antimicrobial resistance. In addition, we have healthcare workers and staff that can transfer it on their hands, scrubs, or personal electronic devices throughout the hospital. And one study found that an antibiotic-resistant bacteria could be isolated in 32% of the sampled clinical personnel's clothing or handheld electronic devices. So as healthcare workers, we have to be particularly vigilant to not be spreading this. So how can we fight this? One of the things that we can do is stress the importance of using antimicrobes just when they are needed, because without improvement of this, by 2050, it's projected that up to 10 million people per year worldwide will be killed by an antimicrobial resistant pathogen, meaning that we have no way to treat them. So what should we do as healthcare workers and patients? Healthcare workers need to follow proper hand hygiene and enforce those contact precautions. They need to limit as much as they can antimicrobial prescriptions unless necessary. Use narrow spectrum over broad spectrum when possible, and then educate patients about the importance of following the drug dosing regimen. For patients need to follow the drug dosing. They need to follow up if it's recommended. They need to properly store medications, and they need to not demand antibiotics from their physicians. So developing new drugs, why can't we just come up with new antibiotics if we have resistance to the ones now? The biggest problems that we run into is that it's very difficult and time-consuming and costs a lot of money. So for every antibiotic that makes it into clinics, there are between 5,000 and 10,000 that have to be screened, and it can take 10 to 15 years and cost nearly $1 billion for each new antibiotic. And in addition, most companies don't want to make new antibiotics because it's only used for 10 days in a patient rather than things that would be used chronically for a lifetime, like high blood pressure medication. So there's very little incentive to come up with new antibiotics. It's often left to these smaller, newer companies. We are looking at the idea that many nations are starting to subsidize that. And in 2012, the U.S. passed legislation to help extend patent rights so companies can actually make the money to recoup the funds that they use to do development of an antibiotic. Some companies are also testing multi-drug approaches with currently available drugs and seeing if you have more than one, could that work? They're also looking at redesigning antimicrobial inhibitors that can block that bacterial resistant mechanisms. And lastly, one of the interesting ones they're looking at is looking at bacteriophages. So viruses that infect bacteria as a therapy possibility. So at this point, you should be able to explain whether or not antibiotics cause resistance in bacteria. You should know some of the ways that humans contribute to the rise in antibiotic resistance. And also, what can healthcare workers and patients do to control antibiotic resistance? And what are the challenges in developing new antibiotics? That's the last of Chapter 15. It's been a great semester. Please let me know if you have any questions.